Okay, let's start. Hello everyone. My name is Isabelle Lefebvre-Vary, and I am the Executive Director of Fertility Friends Foundation. We are very happy to welcome you all tonight. We had a lot of people registering for this webinar, and we know why. We know that Christine is amazing first, but also the topic is very much of interest for people going through fertility treatments, and we don't talk enough about what's going on in a lab. So tonight it's a great opportunity for us to really go through all of that. First, I want to introduce the foundation. This webinar is part of a series called Your Fertility Guide. We launched it in January, and every month we have a webinar on a topic that is of interest to our community. We always try to talk about topics that are not addressed enough, and they are always led by really well-known and renowned experts in Canada. The foundation is Fertility Friends Foundation is a charitable organization. We offer grants to cover expenses associated with fertility treatments, IVF, egg and sperm donation, and gestational surrogacy. We started in 2022, and we already gave over to $340,000 to 70 plus individuals and couples who would not have been able to start or continue their fertility journey without our financial support. As you might know, all of you, fertility treatments are very expensive, and the financial barrier is one of the main barriers. And we're trying to break that down and create a more equal access to fertility care by giving out grants. So tonight, we have the amazing Christine Ye. She is the lab director of Twig Fertility. Twig is one of our partners and is also part of a program that is called the IVF Cycle Program that we created alongside leading fertility clinics in Canada. When we give a grant to a patient of one of these clinics, the grant agreed to cover the rest of the expenses associated with the treatment with exceptions. So you can look it up on our website, and Twig actually was one of the first to join the program and was supporting this program from the get-go. So we're very, very happy and proud to partner with Twig Fertility. So Christine, I'm going to say a few words about you. We're very, very grateful that you decided and you agreed to do this webinar with us. We know how busy you are. So Christine has a deep passion for evidence-based research and an extensive experience in reproductive biology and clinical embryology. Christine has a master's in embryo implantation from McGill University, worked in top Canadian fertility clinics, but also was the head of 2 labs at iGenomics, which I didn't know, and is also advising doctors across North America on how to improve labs and create labs. So Christine designed Twig Fertility's lab based on best practices across North America and with a very holistic and patient-centered approach. And she's an active member of CFS, ESHRE, and the SOGC. And I have to say that SOGC is very foreign to me, but I'm sure it's amazing. A few housekeeping notes. So tonight, you can ask questions throughout Christine's presentation by writing them in the chat. And I will ask your questions along the presentation to Christine, who will answer them as they come. And some of them maybe will answer at the end of the presentation. So without no more waiting time, Christine, thank you again for being with us tonight, and the floor is yours.
Well, Isabel, what a lovely introduction. Thank you so much. I feel very honored to have been asked to come here to present to all of you this evening. Thank you, Isabel, and the Fertility Friends Foundation for thinking of me. I'm very passionate about what I do. So sorry if I go a little overboard, but I'm very excited to bring you all behind the lab door to help understand embryology and your IVF. And as Isabel said, I'm the lab director at Fertility. Most of what you'll see here is from my laboratory because I have access to it, and I have the amazing Naomi who helped take some beautiful pictures to help illustrate to you all. I thought bringing you into the lab, it would be great to show with a lot of pictures and illustrations. So bear with me, but I hope you enjoy it this evening. So first, I don't know exactly what everybody's background is, so I want to go over kind of the basics to set the stage. So we're going to start with what is IVF. IVF is a method of assisted reproduction in which sperm and egg, as you can see here, little tiny sperm cell, big egg cell, are combined to create embryos outside of the body in a laboratory. So that's what in vitro means, in vitro fertilization. One or more embryos can then be transferred into the uterus, or the embryos can be cryopreserved or frozen for future use. So here is a very, very brief history of IVF. So back in 1978, in this month, the first IVF baby was born. Her name was Louise Brown, and she was born on July 25th. So July 25th is Embryology Day, and it's a big event. So I'm really excited to be able to present this to all of you within this month. It's very exciting. Embryo freezing, cryopreservation, started to come into practice in the clinic. So for human eggs and embryos and sperm in the 1970s. It became more popular later on, and it's gone through a whole different phases of what works well. In 1984, the very first baby was born from a transfer with a blastocyst. So we'll get into what is a blastocyst and how does it grow, but this was a really big feat. A big thing that this showed is that the quality of the laboratory was good enough to be able to support blastocyst development, and it also increased live birth rates for patients, which is huge. In 1992, the first baby was born from ICSI, which is intracytoplasmic sperm injection. Again, we'll go into that later, but this was a really big advancement in the ability to provide patients where maybe the sperm quality was very poor, there wasn't a lot of moving sperm, to be able to reproduce with the help of IVF. There's a lot of genetics that happened in between here as well. I don't have that in here, but genetic testing of embryos. And then in the 2020s, we're really excited to start introducing AI and robotics into the laboratories. So here is a little sneak peek into the IVF lab. What is an IVF lab? The IVF lab is a specialized environment that is controlled. It is within the fertility clinic, and it's where in vitro fertilization procedures are performed. The lab's controlled environment and the expertise of the embryologist are crucial and critical to the success of IVF treatment and for making patients dream of having a family become their reality. It's very important that this room stay enclosed, that it's protected. Not a lot of people are allowed in here. You can ask anybody from my clinic. Most people want to go in, most people haven't, because it really is optimized for the eggs, for the sperm, and for the embryos. So what happens in the IVF laboratory? There's lots of things that happen. Most people are a little bit more familiar with is the collection of samples, so collection of eggs, collection of sperm, the fertilization of eggs, and the culture of embryos. This includes egg retrievals, insemination, fertilization checks, embryo culture, and embryo transfers. There's also other highly technical procedures which go on and are conducted by the amazing embryologists that work in this field and have been studying and training and honing their skills for years. Some of these include vitrification, or the freezing of eggs and embryos, sperm freezing, intracytoplasmic sperm injection, or ICSI, as well as embryo biopsies are the most common ones. What people are a little less in the know about is all of the QA and QC, quality assurance and quality control, that go on every single day in the IVF lab. These checks are ensuring that the environment is optimized for the eggs, the sperm, and the embryos that are in culture. So we get to play with a lot of really fun specialized equipment. And I'll just give you a little review. I'll introduce you to some of the equipment that we use. So in this first picture, we have 3 different types of incubators. Incubators are machines that have a set gas level that's different because it's trying to mimic the environment of the fallopian tubes in the uterus. They are used to prepare culture media. They're used to grow embryos. So the three different types that are the most common you'll see is in the back, there's a big box, and that's called a box incubator. A box incubator is a big door. They're typically humidified, which means the relative humidity, there's a lot more moisture in there. It's usually around 80 to 85% to help decrease evaporation. Then in front of that, on the benchtop, you have a benchtop incubator. A benchtop incubator is great because each culture dish or each patient's cohort of samples gets its own little door, a little chamber. So they're all individualized, and you're able to open one and it doesn't affect any of the other ones. So this was a big advancement to be able to go in and out as you need to do to work on the samples without affecting other patient samples at the same time. Now over to the right-hand side, you'll see the big blue box with a picture of an embryo on the front. This is an example of a time-lapse incubator. Time-lapse incubators are phenomenal because it allows you to see the embryo while it's in the incubator. Inside, there's a built-in camera that takes pictures at a regular interval, and you can see the different timing of the cell divisions. You can see different morphokinetics or the movements of the embryo throughout the culture. The embryo goes in at the time of fertilization or just after ICSI, and it stays there for the duration of culture, which lasts between 5 or 7 days. And then you can watch from a screen from your computer, from the front of the incubator, what goes on. And there's different types of time-lapse incubators, just like there are different types of benchtop incubators and box incubators, and they all work very similarly. We also work with microscopes every single day. The samples that we work with are microscopic. You cannot see them with a naked eye, so you need to use different microscopes to manipulate them and to work with them or to process them. Here in the center, you can see, are you able to see my mouse if I move it, Isabel? Yes, okay. So here you can see what we call a micro manipulator or an ICSI rig. There are micro tools that are connected, and this allows us to do procedures such as ICSI and biopsy. And then over here you have a stereoscope, which allows you to move your samples around into different culture dishes, and also allows you to do freezing and thawing in these areas. You can see workstations. We have specified workstations for different procedures that we do. Different procedures are required to be at different temperatures. The workstation also provides another layer of a microenvironment that is controlled for that procedure. It has its own air system that's cleaner even than the laboratory air, which is cleaner than the clinical air. So layer after layer of protection for these samples. And then you also see here, on the white stage here is for identification of the samples. So we'll go into that in the next slide, which is the electronic witnessing system. Most laboratories are now starting to implement different types of electronic witnessing system. And what this does is give an extra layer of security for patient samples. As it comes in, it becomes labeled electronically, so with a barcode on the bottom of the dish, for that patient. So anytime you go to any workstation, the patient's identification comes up within that workstation. So you can see here in the previous picture, we have the witnessing platform, which is where you work with all your samples. And then on the screen in the back shows the patient's information. So it allows the embryologist to know that they're working with the right samples. And it gets down to the point where when you are mixing sperm and eggs, it ensures that sperm is from the partner of the person whose eggs you're working with. So it's just an extra layer of security for both the embryologist and for the patient. We also have now AI-assisted embryo evaluations, which allows the embryologist to help better select the embryos and score them. And then we have amazing liquid nitrogen tanks, which are a very old piece of equipment and work extremely well. I mean, they've been around for years. They're now being improved. We have very large ones, and this is where all the cryopreserved samples are stored. You can see here the vapor from the liquid nitrogen coming off. So let's start with quality assurance and quality control. It's not fancy, it's not fun, but it's critical. Every single day. The mornings always start to ensure that the temperatures are the right temperature, the gas levels are the right gas levels. As you can see here, there's many gas tanks that are hooked up to the gas manifold, which feeds into the incubator. So these incubators are specifically regulated for the processes that they need to do. We like to keep a gas level of about 6% CO2, which is much higher than ambient air, and a very low concentration of oxygen. And again, this is to help mimic the environment of the uterus. And then we have specialized media that maintains a specific pH based on that CO2 concentration. So gas levels are verified every single day. pHs are verified on the regular to ensure that we have the most ideal situation and environment for patient samples. The humidity inside the laboratory and inside of incubators is monitored. All consumables would be plasticware, dishes, tubes, micro tools, all of the lots. So anytime a manufacturer sends a new batch, it's verified to ensure that it's been checked and it's sound and it is not going to hurt the embryos or the eggs or the sperm. We do the same thing with media lot tracking. And then we have the amazing electronic witnessing. We use RI Witness, which uses RFID tags, and it works wonders. So here you just have a little example. Here are some probes that we use. So the incubators themselves tell you what their gas level is and the temperature is. We also use external verification devices to confirm that the incubators sensors are working properly. So there's checks upon double checks upon triple checks. Here you have the gas analyzer. Here we have a little button temperature probe, which is connected to a device, and it reads out what the, here you can see 6.1% CO2 and 5.2% O2, which is perfectly within the acceptable ranges. Here. Now, let's get to more of the fun part. Here, we'll start with the sperm. Sperm collection and processing is a process that we do every single day in the laboratory. While I'm talking, I'll play this little video. On the video is just a microscopic image of sperm swimming around on a slide. Sperm are very small cells. So you have the cell, the sperm cell, which is the head of the sperm. This connected to the head is a flagella, which is a tail-like structure that moves and allows it to swim around. Now, this movement allows the sperm to swim towards the egg, attach to the egg, so the acrosome at the tip attaches to the egg, fuses to it, and then releases the DNA into the egg, which helps to cause fertilization or enables fertilization. So what we need to do in the laboratory, when a sample comes in, typically it's an ejaculate sample, so the sperm is in the semen, which has other cell types and other fluids in there. And what we want to do is we want to clean the sperm. We do that, there's different methods, but typically it's through some sort of filtration. We can use a density gradient, which you can see over on the right-hand side. You have different layers. This is a density gradient that's already been spun. So the semen is put on the top, and then with gravity or a centrifuge, which spins around really fast. If you've ever been to a fair and gone in one of those spinning devices where you feel just pushed up to the side and you feel like you're going to drop down, that's what we do to help clean the sperm. The sperm with a dense enough DNA in its head, as well as the motile sperm, or the sperm that's moving, are the ones that get pulled to the bottom. So you can see here a beautiful striation of the semen at the top. The second layer is more of that debris or extra cells, epithelial cells, or sperm that's not moving, which we can consider dead sperm or dead sperm cells. And at the bottom, this little white spot, that's the sperm. It's called a cell pellet. So they all combine down at the bottom. And then what the embryologist or the andrologist do is they remove all of what we call the supernatant. So all of this fluid up at the top, that gets taken out, and it gets reconstituted into a clean media that allows the sperm to feed. So there's a little glucose in there and allows it to move around. You can then use that sperm for IUI, intrauterine inseminations, for IVF, or for ICSI. So we want to reconstitute the best swimming sperm in the clean media. All the while, we have our RI Witness or electronic witnessing software that identifies each tube that's on our workstation and tells us exactly who the sperm belongs to and who their partner is, so who we can use the sperm with. Now, egg retrievals. Egg retrievals is a beautiful dance between the OR and the laboratory. Our amazing physicians and nurses and sonographers work hard in the OR to retrieve the eggs from the ovaries of the patient who's undergoing the egg retrieval. The follicular, so the eggs are located within the ovary in these little tiny pockets, I guess you could call it, of fluid. Now, the physician goes in with a very small needle, removes the fluid out of these little pockets, which are called the follicles, and that fluid is given to the laboratory in a tube. The embryologist then, at their specialized workstation that's set at 37 degrees, you can see it over in the corner here, takes that fluid, puts it in a dish, and searches for the eggs inside of the fluid under the microscope. Once an egg is found, it's taken out, it's washed, and then put into a culture dish and specialized media inside of the incubators to hold until they're ready to freeze or inseminate. Now, insemination. There's two different types of insemination. We have conventional IVF, or conventional in vitro fertilization, and then we have ICSI, intracytoplasmic sperm injection. So we'll start with conventional IVF. With conventional IVF, you take your eggs and you put them into a dish. To that dish, you add a specific quantity of sperm. The sperm then swims with that flagella tail around in the dish until it finds the egg. You can see here, it will attach to the zona, which is the outside of it's the shell essentially. And once one starts to bore its way through the zona hardens and it doesn't let any other sperm in, because you only want one sperm to fertilize your egg. And the eggs have the innate ability to prevent any other sperm. So it really is the fastest win. You want to get there, attach, get in there. Then once the sperm gets in, the acrosome helps it fuse to the top of the cell membrane of the egg, which I'm pointing to here, and it releases its DNA. So if we go back, this part fuses to the outside of the egg, or the membrane, and then it releases its DNA, if we go forward, into the egg. At the same time, it activates the egg, turns the egg on, and says, hey, let's continue this process to get fertilized. The next day, the embryologist looks at the eggs at a specific time, typically between 16 to 18 hours after fertilization, and looks for these two morphological features, which are like little tiny round bubbles inside, which are called the pronuclei. One of them is from the sperm, and one of them is from the egg. And this is the sign of fertilization that the embryologist looks for to note or determine if an egg has fertilized or not. And this is a very neat picture because you can see all the sperm that's attached to the outside that did not make it in to fertilize, and that one lucky sperm got in there and fertilized this egg. Now, the other type of insemination is ICSI. With ICSI, the embryologist, you can see in this picture or this video that I'm playing, will search for the best sperm under the microscope. So they're using this invertoscope here with the microtools. They kink the tail, because kinking the tail activates the sperm, which then will activate the egg. You can see Alana here doing the ICSI. Then once it's kinked, they'll pick it up into this microtool. You can see it's got a little beveled head to help break into the embryo, or the egg, excuse me. And here you can see that sperm is then injected directly into the middle of the cytoplasm of the egg. And here is just a blow-up picture. You can see the little tiny sperm here. This is a great illustration of the different sizes of the cells. So an egg cell is very large compared to the really tiny sperm cell here. So that sperm cell is then injected into the egg. Just because the sperm goes inside of the egg does not guarantee fertilization. There still has to be activation of the egg, activation of the end of meiosis, essentially, to be able to create a zygote, which is a fertilized egg. So once you have your fertilized egg the next day, you can see that here in this picture, then we get into the embryo development and culture portion. This lasts for 5 to 7 days, where one cell becomes over 300 cells in a blastocyst. So on day 1, the embryologist does observations and ensures or verifies which eggs have been fertilized or not. So which eggs show or are you able to visualize the two pronuclei in. One pronuclei contains the DNA from the sperm, and the other pronuclei contains the DNA from the egg. The cells then start to divide. They divide into 2 cells, and then they divide into 4 cells. That's typically on day 2. By day 3, we expect to see between 6 and 8 cells. Also on day 3, this is the time point at which the DNA from the sperm starts to actually contribute to the development of the embryo. So up until this point, it's the DNA from the egg and mitochondria, typically from the egg, that's supplying all of that energy and ability to divide. On day 4, all of those cells start to merge together, and you stop being able to see these very clear cell divisions, or the divisions between the 2 cells, and it becomes what we call a morula. Now, there's still a lot of cell division and a lot of action that's happening in here. A big portion of this process is when the cells start to differentiate into 2 different cell types. So up to this point, the cells are all the same. At this point, there's a subset of cells that become the trophectoderm. So I'll point to it here in the blastocyst, which are the outside cells within the blastocyst or the embryo. That's what will attach to the uterine lining at the transfer and create the placenta on the side of the embryo. And then the other type of cell is the inner cell mass, or the ICM, which is this group of cells on the inside. And those are the cells that will eventually become the fetus. So after day 4, between day 5 and day 7, the blastocyst develops. A blastocyst is an embryo that has a fluid-filled center, the blastocele, with cells all around the outside and the inner cell mass on the inside. And I like to equate it or take the analogy of a soccer ball. So it's almost like it blows up into a soccer ball, and as the cells divide and there becomes more, it gets larger and larger and larger until it gets to the point where it hatches. It has a little break in the zona, it hatches out of the zona, at which time it would attach to the uterine wall. So these steps, what we're able to see in vitro or in the laboratory, are the same steps that happen during natural conception. While the egg is released from the ovaries, it gets fertilized at the top of the fallopian tubes, and as it comes down, it goes through these processes, and by the time that it reaches the uterus, it will be a blastocyst. So it's the same process externally as it would be internally.
Christine, we have a question.
Yes, please.
So the question is, does this mean if the development arrests after day 3, that it is possibly a sperm issue?
Yes, it could be possibly a sperm issue. There are many different variables. It's always best to speak with your physician and take in all the information around to determine whether it is a sperm issue or not. But yes, it could be a sperm issue if you have day 3 cell arrest.
Thank you.
Great question. Some laboratories now have started using AI in the laboratory, and I alluded to it earlier. AI, there are different databases that are coming out, or companies that are coming out, which add an additional layer. Well, I guess I'm going to go back. I'm going to go back a step because I forgot to explain how blastocysts, or these day 5 to day 7 embryos, are graded or prioritized. So traditionally, it's what I like to call a beauty pageant. We look at the number of cells, we look at the quality of the cells, the expansion of the blastocyst, and we give it a grade. One of the common grading systems is called the Gardner grading system, where it's a number and 2 letters. The number is between 1 and 6, and it alludes to how expanded the blastocyst is. And then there's 2 grades. The first grade, A to C or A to D, depending on the laboratory, is the grade of the ICM. So you're looking at here, A is for awesome, B is for beautiful, C is for it's all right, and D is usually very low quality or non-existent if you're looking at an A to D separation. And then the trophectoderm cells is the second grade, which also goes A to D, same type of grading. So the embryologists are very skilled at looking at the embryos and determining, and then based on the grade, you're able to prioritize which blastocyst to transfer. Now, AI brings a whole nother layer to that because it allows AI systems to look at either pictures of blastocysts or the whole video of the full culture of the embryo, from fertilization to blastocyst and help determine which embryo has the highest potential for implantation for pregnancy and potentially live birth, depending on how the algorithm was trained. Is the system perfect? No. Does it still need embryologist input? Yes. But together with the embryologist grading and the AI, it can augment the ability to choose the best embryo or prioritize the best embryo for transfer. So here is just an example, and this is also what we would see in time lapse. So you can watch the embryo fertilize, go through its cell divisions, and eventually it gets to the blastocyst stage. Here you can see this AI program is grading these different embryos. So all embryos, these 4 embryos may look the same or have the same Gardner grade, but then that AI score can help choose out of these beautiful embryos, which one do we choose to transfer? Which one is it ranked? For example, this embryo you can see here has a score of 10, but it's at number 3. So there's 2 other embryos in this cohort that the AI system would suggest transferring first. One thing that we are very fortunate, we do use AI in our laboratory, and we have the ability to provide a live stream of the patient's embryos and culture to the patients at home. So for all of the eggs that are fertilized, the patients get a video and they can watch them throughout the duration of the culture period. Isabel mentioned before that we try to take a very holistic approach to IVF treatment. A big part of that is giving patients information and a touch point in with the clinic, making sure they understand what's going on and educating them. And we feel that in this day and age, we have the technology to be able to give them sight into their embryos. So they're going to have all that information anyways. It's been phenomenal for us so far. And I think our patients have very much enjoyed, maybe not enjoyed, fertility. I don't think anybody enjoys. Nobody wants to be here, but they have appreciated the ability to feel a little bit more part of the process. And if we do have any patients out there who have had the ability to live stream their embryos during culture or haven't, we'd love to hear your feedback and hear how you've been experiencing it. Because obviously this is coming from the laboratory side of things.
I have
but also
for you, Christine. I'm so
sure.
sorry to interrupt.
No, don't be sorry. It's great. Interrupt anytime.
It's fascinating. In the video that you just showed, it's a question from me actually. In the video that you just showed, there on the left side, there is some kind of bubbles. What is it?
Oh, that's a great question. So when an egg is retrieved, it's surrounded by a bunch of cells that are called cumulus cells. Cumulus cells are maternal cells that are like nursing cells. They provide nutrients and different paracrine factors that help the egg. And it's naturally occurring, and they're all surrounding the egg. At the time of fertilization, or just after fertilization in the laboratory, we remove all of those cumulus cells. Now, sometimes a few cells stay on the zona. They're a little bit sticky, and we don't want to be too rough on the egg. So that is just some of the leftover maternal cumulus cells that are attached on the outside of the zona.
I see. Okay.
Great question.
Thank you. Other question. If you have a time lapse of your embryos, can you run it through an AI program for further insights?
In theory, yes. It would depend on the AI system. Some AI systems actually just are based on a single picture of an embryo. Others take the time-lapse video. If you have a time-lapse video and you're able to export it in a specific file type, and the AI system or company that you're working with are able to import it into their system, then in theory, yes, you could.
Thank you.
So theory, yes. Practicality and actuality, maybe, maybe not. Depends on the situation.
Okay. Hold on, because I see a few more questions. To the person named E, I will answer in the chat directly. And Regina, for embryo freezing, which of the 2 methods are commonly used? ICSI or natural fertilization? If natural is used first and wasn't successful, with the lab turn to ICSI method immediately?
That's a good, Can you repeat the beginning of that question? Was it regarding embryo freezing?
Oh, yes, embryo freezing.
Okay, so embryo freezing occurs whether IVF or ICSI is used. So once you get to the blastocyst stage, any embryo that reaches the blastocyst stage will be frozen, if not transferred fresh. So there's no differentiation between that. In regards to if IVF fails, is ICSI used? It depends on your laboratory and it depends on the situation. In some laboratories and in certain situations, yes, ICSI would be used if there was what we called failed fertilization. Typically, there's a certain percentage of eggs that would have had to fail fertilization, or sometimes all of them, and then rescue ICSI is performed. It's not a guarantee that fertilization is going to happen. There's lower chances of fertilization, lower chances of blastocyst development, but it can work. I always consider it kind of a Hail Mary. You have all the samples there, why not try it? But there's no guarantee, and likely it's not going to produce much, but it is done in some laboratories.
Thank
I
you.
hope that answers your question.
I think so.
Anything else? No? Okay. So another thing that AI has been able to enhance is the programs are typically able to produce really nice reports that are very beneficial for the physician as well as the patients to review at the review appointments, to look at what embryos have been transferred, what do they have frozen and ready to use in the future, to evaluate how their cycle has gone. So embryo transfer. We're all here for the embryo transfer. We want to choose the best embryo, as we've discussed, transfer it into a uterus that's ready to receive that embryo, and try to provide a pregnancy or help to create a pregnancy for these patients. So the blastocyst is carefully loaded by an embryologist into a very small catheter with a little bit of media in there. The blastocyst itself, or the embryo, sometimes there'll be a day 3 transfer, rare, but sometimes. So the embryo is surrounded by a little bit of fluid, and the fluid is surrounded by 2 little bubbles of air. Now, the bubbles of air are important because once the catheter is transferred to the physician will insert that catheter, you can see it here, this white line, into the uterus. So this balloon-looking structure in this ultrasound is the uterus. So the catheter is entered in, and then the embryo is what we call is flushed into the uterus. So that media with the embryo is put in there, and the little air bubbles allow the physician to visualize the embryo leaving the catheter and going into the uterus. And they want to make sure that it's in a certain part of the uterus. So this little the air bubbles show where the embryo is placed in the uterus. And then about 2 weeks later, the patient undergoes a pregnancy test, which we all hope is positive. And then in around 7 weeks later, an ultrasound is used to confirm a viable pregnancy if the original pregnancy result is positive. So here you have a beautiful blastocyst. Here's the ICM here, the trophectoderm cells loaded into the catheter and transferred into the uterus.
Amazing. We have a few more questions.
Great.
If you retrieve an immature M1 egg, what is possible to try to get it to M2 in a lab?
So there's a process called IVM, which is in vitro maturation, in vitro in the laboratory, maturation, maturing an immature egg to a mature egg. Some laboratories will do this. It's another technique and process that is part of some laboratories. Once it's matured in the laboratory, then it can be injected with sperm and hopefully fertilized. So yes, that is possible.
If traditional IVF is used instead of ICSI, is there some advantage provided to the oocyte by allowing some time for further maturation post-retrieval?
Yes, in theory, the embryo or the eggs might mature from the time of retrieval, and they're surrounded by the stimulus cells until the time that the sperm attaches into the egg and inseminates it. That is possible.
Does Twig perform IVM?
For select cases.
Okay.
Yes.
That's all for the moment.
Great. So I see the time. I'm going to try to get through these next slides, but if we don't, that's great. I love the conversation and I hope you're all enjoying this. So another technique that we do, and I'll start playing the video and you can watch it as I'm talking, is embryo biopsy. So the embryo biopsy is performed to retrieve 4 to 6 trophectoderm cells, so those outer cells, to then send to a genetic lab for testing. What the genetic lab can say or tell you from their analysis is if the embryo has a sufficient number of chromosomes. They don't have too many, they don't have too few. Chromosomes are structures of DNA. Every cell should have 46, and this testing ensures that those embryos do have the correct number. It can also detect if there's an incorrect number. Certain conditions everyone is very familiar with is, say, for example, trisomy 21 is when you have too many chromosome 21. So you'd have 3 of them instead of the 2 that you typically have. So here in this picture or this video, you can see this is a microsurgery essentially that the embryologist is doing. They take a few cells here and using a very small laser cut between the cell junctions to remove a biopsy or a sample of cells from the blastocyst. The blastocyst itself is unharmed. It then gets vitrified or frozen and conserved for later, whereas the biopsy cells, which are removed in the biopsy needle here, are put into a really tiny tube called the PCR tube. It's frozen down and sent to the genetic lab for testing. And then the results will help to determine which embryos are most suitable for transfer. So the normal embryos that have the right number of chromosomes are typically the ones that are prioritized. And so here you can see the little clump of cells that would have then be sent to the genetic testing laboratory. And here's just a fuller view, another embryo that's being biopsied. Here you can see there are more cells that are outside, but only a few will be taken. And we use the same ICSI rig or micromanipulator to perform it. So after biopsy, or if no biopsy is being performed, but we're freezing down the embryos or eggs, you can freeze eggs as well, it's done at one of the IVF workstations. First, what happens is we identify each individual device. So we know exactly which embryo goes on which device. We only put 1 per device. Excuse me, this device is very small. You can see Deborah here, who's one of our amazing embryologists. She's now working sample processing. She's taking that embryo and putting it through different solutions to help dehydrate the cells, because you want to remove as much water as possible, and then you put cryoprotectant inside of the cells. Once they're ready, they are then loaded. You can see it here on a very small device. This is the embryo itself. Here is the device, and it's within a fluid that's called vitrification solution. And then it's submerged in liquid nitrogen and vitrified. What vitrified means is to make glass. And essentially, you want to freeze the cells in a glass state, and it freezes them. It stops all of the action that's going on inside, freezes all the organelles, and kind of puts it in a pause. Embryos and eggs can be frozen for a long time. They're always submerged in liquid nitrogen and stored. And then at the time of warming, you go through another process to rehydrate those cells and reactivate all of the action that happens within the cells. So here is an example of what our cryo storage looks like. You can see here, there's many different what we call canes. These are the top of them, and then the rest of it is completely submerged in the liquid nitrogen, which is at negative 196 Celsius. Each individual cane is per patient, and their samples are inside of the cane. So there are multiple devices. The devices are what the embryos are on inside of a cane for every specific patient. And there's a whole cryo inventory process. These storage tanks are monitored 24/7. To be honest, all of our equipment is monitored 24/7 to make sure that everything is working perfectly all the time. The other thing, we're going back to QA/QC. Outcomes are always monitored as well. So the lab has KPIs that we look at to ensure that the culture system is in check. Everything's working properly, to ensure that the equipment is functioning properly. Because if there's any malfunction or issue with any of these steps, it could affect the fertilization rate. It could affect the blastocyst rate. It could affect the pregnancy rate. So these are metrics that are always reviewed and viewed again to ensure that the embryologists are performing at the top of their game, that the equipment is performing exactly the way we need it, and to ensure that there's security and peace of mind for both the staff and the patient. So again, the other thing that we always do is we have fun together and we celebrate each other. Here are some great pictures of our amazing staff at Twig, our lab staff, and they work extremely hard and they're amazing, just like other embryologists all around the world. I have so much respect for each and every one of you. You do amazing work and we help patients in ways that just make such a difference to their lives. So very difficult work, very small work, very just amazing things that we do.
Amazing.
So thank you everybody for joining me this evening and indulging in what it's like behind the scenes in the IVF lab. So if you have any other questions, I'd be happy to answer.
Yes, we have a few more questions.
Great.
For women over 45 using her eggs and or donor eggs, how many rounds of IVF until success on average?
That is an excellent question that should be asked to your REI or your physician.
If you donate
There are many different factors. That's all very personalized. So I wouldn't be able to just make a statement.
If you donate an embryo for research, what usually happens to it?
That's a great question. So embryos can be donated for different reasons. They can be donated for research, which would then go into a research project. Maybe a new AI system is coming out. Maybe there's a new technique that needs to be explored. For example, there's been a study where they wanted to correlate the chromosomal structure between the ICM cells and the trophectoderm cells. So some laboratories underwent a study to then take an embryo and dissect it into multiple parts and do genetic testing on the different parts and see if they all corresponded together, if they had the same results or not, to make sure that the trophectoderm cells was truly a good representation of the ICM, which would eventually become the fetus. So that's an example of research. Embryos can also be donated to the improvement of procedures. That would be, for example, if the laboratory wanted to implement a new procedure, you can't just implement anything. You have to do a lot of testing and validation. So donated samples are great to be able to say, introduce a new vitrification or freezing technique for bettering the practice and the outcomes. You don't want to use a new technique on patient samples, so you want to start doing it on donated samples. You can also donate your embryos for use of training. So embryos that are donated for training are then used to train up new embryologists or train up seasoned embryologists in new techniques. So it gives samples. Everyone needs samples to learn on. So we'd use those samples that are donated for training for that specific reason. And usually it's detailed within the consent form and you can choose which of those options you prefer.
Thank you. Are frozen embryos stored in the lab or off-site?
It can be either or. At Twig Fertility, they're stored on site. Some other clinics have off-site storage. Sometimes it's within the same company. Other times it's a cryo storage facility that stores the embryos off-site.
Is calcium inophore, I hope that I pronounce it okay, not well, but okay, used in the culture media in all situations?
I feel like we have some people who know a little bit about embryology listening in. Calcium ionophore is not used in the culture media. It's a technique that can be, the calcium ionophore is introduced to the egg at the time of fertilization and only at that time. It's used to activate the egg. And it's not used for all cases. It's used for very select cases, typically when there's total failed fertilization from ICSI.
Thank you so much.
Mm-hmm.
What systems are in place to ensure you are using the right embryos for the patient?
That's a great question.
Yeah.
So I talked about the electronic witnessing system. That is on top of all of the checks and double checks and labeling that's naturally done in the embryology lab and it's been done for years. So all samples are specifically labeled with 2 patient identifiers at least, as well as the date that the procedure was performed. So then you have a date, patient, sample, and the sample number. this is written and identified on any device or container that the sample is in, the embryo is in. On top of that, with the electronic witnessing, any sample that's frozen is barcoded. So scanned, barcoded going in, it knows exactly which embryo it is, what patient it belongs to, the date that it was frozen, the time that it was frozen, the IVF workstation that it was frozen at, the embryologist that froze it. All of that information is stored within that barcode. Then at the time of thawing and transfer, if you're doing a frozen embryo transfer, at the time of thawing, the barcode is scanned and it pulls up all that information automatically. That information is then verified by 2 individuals, and as well as all of the written information that's on the device as well.
It seems very thorough.
Yes.
Yeah.
It's phenomenal. I love it.
Great. We're at the end of the webinar, Christine. It was fascinating. So thank you very, very much for putting this presentation together, for the videos also. That was truly amazing, very interesting, and thank you so much for bringing us all behind the closed door of a lab. It was really. I feel a bit special tonight, so thanks to you. So thank you so much. Oh, I have one last question for you, and I think we are going to wrap up after that. Are antioxidants or any supplements ever added to the culture medium?
So it really depends on the laboratory procedures, and the specific case. I've worked in areas where in certain situations, certain things are done for that patient. IVF is very personalized. And the physician in the clinic that you go to is specialized and will provide you with special care depending on what you require and what you need. So it's case by case.
Okay.
Mm-hmm.
Thank you so much again. A few things. We have actually a World IVF Day giveaway on our social media. So you can go to our Instagram account and look at the post that will tell you all about the giveaway. It's for $250 gift certificates to leading Canadian brands, wellness brands. So there are some rules to enter, but we thought that it would be nice to do something for World IVF Day on July 25, as Christine mentioned at the beginning of her presentation. If you are interested in our services, you can also register to our e-newsletter, follow us on social media so you know when our application platform will be open next. It's going to be on October 1st, and we are going to be open for a month to receive applications, grant applications, and the grants will be disbursed at the beginning of December. So again, thank you so much, Christine. Amazing webinar. And I hope to see you soon. And good evening, everyone.
Yeah.
Thank you.
Thank you. And everyone should tune in to Dr. Nayat's presentation. I believe it's next month on different technologies inside the clinic. It's probably going to be phenomenal. So thanks again, Isabel, and thanks to all the amazing work and support that the Fertility Friends Foundation provides for patients.
Thank you, Christine.
Thank you.
Have a good evening. Bye-bye.
You too.
Bye.
Bye, everyone.